Jen, we're good, I think.
Why can't I not?
Are you looking for the pointer, the red one?
I have the pointer. You should see the pointer, right?
Yeah, we can.
When I was trying to, you're recording this, and I was trying to acknowledge this, and I had to get out of PowerPoint back into Zoom.
Ah, okay.
And I have a fire drill. I'm not sure. I hope I can stay in my office.
I hope it's not a real fire.
No, it's not real. It's a drill, but they sometimes have you go out of the office.
Yeah.
Okay, we're live on YouTube. We'll just get started in a minute. I want to we had a bunch of signups at the last minute, and because we don't have like the fancy Zoom that does auto emails, I'm going to see if I can send a last minute reminder email so that those new folks can get the Zoom link. So give me 1 second to do that. Maybe Ankita, I'll send you something.
Okay.
Okay. You know what? I'll send that last minute email after we get started, because I don't want to delay things as folks are continuing to drop in. Always during the first 5 minutes, we'll have a a lot of changeover. So yeah, let's go ahead and get started so you can have maximum presentation time. So hello, everybody. Welcome. I'm sorry if you can hear in the background, my cat is doing that thing where he sings the song of his people and walks around yowling. So apologies for that. Anyway, so today I'm delighted to welcome Professor Holger Knaut. And Holger did his graduate studies with Dr. Christiane Nusslein-Volhard, and he went on to do his postdoctoral work with Dr. Alex Shire at NYU and at Harvard. So Holger is currently an associate professor of cell biology at the NYU School of Medicine. You can see him sitting in his office at NYU, where they keep the air conditioning on in late October. And so I was especially excited when we were able to schedule Holger for the series, because I actually realized this summer, Ankita and I were talking about this, that we've had a real lack of zebrafish people over the past couple of years at this seminar series. And we've really had a lack of people studying the lateral line, which, as I think we'll see today, is one of just the absolute coolest cell migration systems. So with that, I'll turn it over to Holger, and just want to say thank you so much for being here. Thanks everybody for attending, and I'm excited to hear about lateral line cell migration. Thanks so much.
Okay, good, thanks. I mean, I don't know how you do this. Can I take questions as I go, or do you want me to take them at the end?
So we usually have, sorry, go.
Usually
we have questions at the end, but if there's any burning questions, we can have that during talk as well.
I'm okay being interrupted.
Okay,
so,
perfect.
What I'm going to do is I'm going to talk to you about tissue motility, and I'm going to give a brief introduction to the model and to the background that you need to know in order to understand some of the rationales of the experiments. And then I'm going to tell you a little bit about how a tissue, what the motor in the tissue is that we're studying, the primordium, what the substrate is that it's using to crawl forward, and then how it couples its forces to the outside, and how the outside is being pushed and pulled on by that tissue. So the tissue that I'm talking about is as you heard already, the posterior lateral line in zebrafish. So this this tissue is born behind, so this is a cartoon of the embryo, right? This is the eye, this is the tail, and here, this is supposed to be the ear, and just behind the ear, you have a tissue of 140 cells that is born at about 18, 19 hours post-fertilization, and then over the next 20 hours, it's going to migrate along the body to the tip of the tail. So it does so because from its back, this tissue is depositing sensory organs, and they will sense water flow. We use a system to understand cell migration, tissue migration, because the tissue is migrating at the very surface of the embryo, just under the skin. And you're going to see a lot of this today. So you can image it at high resolution, both temporally and spatially. This is low resolution, and you see this tissue migrating over the trunk and the tail, what you saw in the cartoon before. And what you notice is that it's pretty much steadily migrating. It's like fairly constant speed, fairly constant in its directionality. And what I would like to talk to you today about is how these cells actually physically push and pull themselves forward. So that's basically the nutshell topic of today. What you need to know for some of the experiments is how this tissue actually knows where to go to, so the directionality. So there are three factors which are important to give that tissue its directionality. And when you mutate them, so this is a wild-type embryo, right? Eye, tail, and here in green with GFP labeled is this tissue, the primordium, and two of the sensory organs that it has already deposited. If you mutate one of these factors, it's called CXCR4. It still moves. Motility is not impaired, but directionality is impaired. So it's basically the cells are swirling and not getting anywhere. Same is true if you mutate another factor called SDF1A or CXCL12. And the third factor is CXCR7. So these genes are encoding different things. CXCR4 is a GPCR, a G protein coupled receptor, that binds to a chemokine that is SDF1A or CXCL12, the other factor, the second factor. When the chemokine binds to the receptor, you elicit G protein coupled signaling, and that's the receptor that endows the tissue with a sense of directionality. So where higher chemokine concentrations are, that is being detected using CXCR4. The third factor is also a chemokine receptor, but an unusual one. It binds to the chemokine with higher affinity than CXCR4, but it doesn't elicit signaling in the system, but it takes a chemokine, internalizes it, targets it for degradation, goes back to the membrane, grabs the next one. So it's a negative regulator of the chemokine signaling system, and you can think of it as a clearance decoy receptor, like a vacuum cleaner for the chemokine. So if you put these three factors together with their expression domains, the chemokine, the guidance cue, CXCL12 or SDF1A, is expressed all along the migratory route. The tissue that's sitting on top of it is expressing the guidance receptor, CXCR4, and the clearance receptor is made only in the back. So it has a higher affinity for the chemokine, so it outcompetes CXCR4, takes it up, internalizes it, and degrades it. So what that does is, across the tissue, in terms of attractant concentration, you have attractant made at the stripe, cleared in the back, that will give you a gradient across the tissue. And because the tissue is using this gradient to crawl forward and has a vacuum cleaner for the attractant in its back for the chemokine, it will constantly make its own gradient as it migrates. So this is as much of an introduction to the system I want to give you. And I'm going to briefly talk about the motor in the cells, or at least as much as we know about it. So in single cell migration, the model is, or a lot of studies show that you have CDC42 in the rack in the front and row A in the back. These small GTPases regulate actin, and we were looking which ones of those are asymmetrically expressed in the cells, in this tissue, in the primordium. We couldn't find any polarity in CDC42 in the rack, but we did find, when we looked at row, active row, using AHPH as a sensor for it, a localization-based sensor, we could see that in the primordium here, this is the front, this is the back. We saw that there's a lot of localized row A activity based on the sensor, and most of that is actually on the basal side of the cells. And when you block row A signaling, the dots go away. If you overactivate it, you get more of them. Okay, so briefly, what does row A signaling do? Row A is signaling through a kinase rock that then activates myosin 2, that then contracts the F-actin network. So you get actomyosin contraction. You can manipulate this pathway at different levels. I'm going to tell you about 3 things we did. we used a inhibitor against ROA called C3. We used a activator for ROA, GEF that turns on ROA, and we used a phosphatase that's always on to dephosphorylate myosin, so you always keep it in the inactive form. We wanted to do this because this is important in many cells, right? So we wanted to only turn this on in the migrating tissue during migration. So the way we did this is we made use of a hormone-inducible system called the LEX-PR-LEX-OP system. Briefly, how this works: you put LEX-PR, the hormone receptor, under your favorite promoter. In our case, we're using the guidance receptor that is expressed in the migrating tissue. And when you then give the hormone, the hormone receptor gets dimerized, binds to a little sequence, LEX-OP, and then turns on the transcription of whatever you put behind the LEX-OP. So in our case, we put C3 and the other two things behind it. And I'm going to show you what happens if you then express C3 in the migrating primordium only. So the way this experiment is going to work, my computer works, yeah. You're going to have the embryo, you're going to have a marker for the migrating tissue, and you have in the fish LEX-PR expressed in the migrating tissue. And when you add the hormone, you make your inhibitor of ROAC3, and you can ask, is that affecting the migration of the structure? So is something changing? So when we do this, what you're going to see is that on top is a control. So where we did not induce C3, the inhibitor for ROA. If we induce C3 in the tissue, only in the migrating tissue, so the skin and the muscle underneath are all fine, you slowly, the tissue slowly starts to cease migration, right? So you stall it. Okay, so that was, and this is also true for the RGF. If you overactivate it, it's also true if you keep myosin 2 in its inactive form. So if you look at the pathway, what Rho does is, or at least one of the main things it does is it leads to actomyosin contraction. So we're wondering whether actomyosin or actin, F-actin, is affected when we do this treatment. And when you look at the very tip of this tissue, and you look at actin using F-tractin as a marker, if used to M-cherry, and you image that, you're going to see that there's a lot of dynamic of actin, right? You have a lot of actin moving backward, flowing backwards retrogradely in the cells. If you impair the pathway by putting an inhibitor on, you block most of this dynamics of the actin network. Okay, so probably what's happening, ROA is inducing actomyosin contraction in the cells, and that is important for them to move. So that probably is kind of like the motor in the cells. So if you were to think of like yourself, you're walking, ROA is basically telling your muscles to contract, right? So your legs are moving forward. Okay, so that's as much as I want to talk to you about the motor. So if you think of like, somebody walking, you need a substrate to walk on. For example, if you walk on the beach, you need the sand to walk on. We need to know what these cells are walking on. And in cell culture, you normally know this, right? You have like your cell, you have integrins that are coupling the substrate to your actomyosin network. And what we didn't know is what is the substrate in the embryo for these cells when they crawl forward. So the first thing we did to kind of get an idea of what the substrate might be is just electron microscopy. So we took the embryo, did a cross-section across the primordium. So this is a cartoon of what this looks like. You have the outside, the skin, and then here the migrating tissue, and underneath the muscle. So if you do an EM, you see the skin here. You have pseudocolored the cross-section of the migrating tissue, and then underneath it, you have the muscle. So if you zoom in this part here, you actually see that there is a thin basement membrane that's about 200 nanometers in thickness. So that was like a EM idea of what might be the substrate. We wanted to get molecular evidence for this. So the basement membrane components, molecularly, the core components are collagen 4. laminin, prolyl, nitrogen, and we used laminin and to look at this basement membrane in the fish embryo by tagging it with GFP. Okay, so this is basically laminin, gamma lamin or lamin C1 in fish labeled with GFP. And when you now look at where the primordium is, you see that the basement membrane is around the primordium. This is the front. Again, this is the back. You see a bit of the skin here. This is the muscles. Those are the myotendinous junctions. If you do a cross section, you see the basement membrane. The skin would be or is on top of this embryo here, and you see underneath it the basement membrane, right? And if you look at the front here, you see the basement membrane. The skin is on top. The primordium is actually pushing most of the basement membrane towards its basal side, towards the muscle, and the skin is going to stay on top. So what it does is it seems to wedge between the skin and the muscle and pushing the basement membrane, separating the basement membrane from the skin on top and pushing it towards the muscle, okay? So the idea was maybe the basement membrane is one of the substrates that these cells use to crawl. Okay, so this is a cartoon version of this. You have the basement membrane underneath it, you have the cells of the primordium, you have the skin on top, and most of the F-actin, the filamentous actin, is localized to the basal side of these cells. I didn't show you that, but that's also true. Okay, so the question was, do we or do the cells need the basement membrane to move? One way to do this is to just impair the basement membrane. Ask, is that impairing migration, right? So we did this. It's a very simple experiment. You take a laminin C1 mutant. So laminin is a trimer. So we killed one of the components. And when you look at collagen 4, you see that you are disrupting the basement membrane severely, and you also stall primordial migration. The problem with this approach is that you also kill the production of the attractant, right? So here is a wild-type embryo, and you see the mRNA for the attractant, the chemopen SDF1A, and this is here where the primordium is starting to migrate. It's going to migrate along the trunk and tail in a basement membrane. Well, in an embryo where the basement membrane is impaired, you do not make this attractant stripe, so you will not be able to migrate anyways, right? So to figure out whether the basement membrane is required, we needed to use a complicated genetic trick. So the way we did this is we wanted to create a scenario where we would take away the endogenous attractant, the chemokine, put it back in the muscle so we could make it, and then do this in an embryo where we have the basement membrane and an embryo where we don't have the basement membrane, okay? And ask whether we can still lure the structure towards the muscle if the basement membrane is not present, okay? So the way we do this in fish, we inject either this is a muscle promoter using M-Cherry, or using a muscle promoter expressing the chemokine in the, from the muscle, getting this in the muscle of the fish, right in front of where the primordium is, where the tissue is that is attracted by this chemokine. We do this in a chemokine mutant, so there's no competition from other sources in the embryo, and then we're going to do this in an embryo that is wild type for laminin C1 and one that is mutant. So in this case here, we're not going to have a basement membrane. Okay, so when you do the control, you have a fish that doesn't make the chemokine, the attractant, the primordium does not move. If you give back the chemokine in the muscle, you are attracting the tissue. So migration works again. So the system in principle works, right? So if you now do this in an embryo where the basement membrane has been compromised because we mutated one of its components, LAMC1, you cannot do this. So this is the control where we just express M-Cherry in the muscle. That shouldn't attract the tissue, and it does not. But if we now put the chemokine in and we don't have the basement membrane, we cannot recruit the tissue to the source of the chemokine. Okay, so there's one caveat here. The problem is that we do not know whether the attractant can signal and diffuse in the absence of a basement membrane. So we repeated this experiment where we did the same thing as before, but we asked whether the primordial cells can, in the absence of the basement membrane, still see the chemokine. So for us to be able to see whether the cells can see the chemokine, we used a sensor for the chemokine that we developed a while ago. So the way this works is we're expressing an RFP fused to the chemokine receptor CXCR4 from the same transcript. We're using an internal ribosomal entry site, and we're expressing membrane GFP. Okay, so that what that gives you on the cell is it's going to be red for the CXCR4B RFP, and it's going to be green for the membrane GFP. So overall, the membrane is going to be yellow. So when the chemokine comes, it binds to the chemokine receptor. And the nice thing about this chemokine receptor is it gets internalized. So it's getting stripped from the membrane. So your RFP is going to be removed from the membrane as the chemokine binds to the receptor. So the ratio on the membrane of the green to the red on the membrane tells you something about signaling. So if you have low signaling, very little chemokine, it's going to be more yellow. If you have high signaling, you're going to get a green membrane. So we did the same experiment as before. We used the chemokine mutant. We put either just mCherry in the muscle and ask whether we can induce internalization of the receptor. We cannot because there's no chemokine. That makes perfect sense. The CXCR4B here in magenta is staying on the membranes of the primordium. If we do the same thing and we're expressing the chemokine, the receptor gets internalized. Okay, so that makes sense. The basement membrane is there. The primordium can see the chemokine. If we do the same thing in an embryo where we don't have the basement membrane, we can still induce internalization of CXCR4. So you can see this here, right? So this is a double mutant, doesn't have endogenous chemokine. We make the chemokine in the muscle. The basement membrane is compromised, yet the chemokine still gets to the tissue and still induces internalization. So diffusion and signaling of the chemokine does not require the basement membrane. So we conclude from this experiment that actually we need the basement membrane in order for the structure for the primordium to migrate towards the chemokine source. Okay, so that is basically how we identified the substrate, the basement membrane, to be required for efficient migration of the primordium. So what I talked to you about is basically like a person like walking, right? So you have row A that induces your contractions of your muscle. You walk on the beach. The sand would be the basement membrane for us. And then you need to, when you walk on the beach, you need to actually couple the force that you're generating to your substrate, to the sand, right? So we need to couple the force, and we were wondering how this is working in the primordium. Again, in tissue culture, often this happens by focal adhesions, so integrins. And what I'm going to do is I'm going to tell you about integrins in the primordium. And we did the same thing for talons, but I'm not going to talk about it just for the sake of time. So to first figure out whether integrins could be involved, we asked whether integrins are made in the primordium. To do this, we knocked in GFP into the integrin beta 1b locus. In fish, integrin beta 1, there are two versions of it, 1a and 1b. So we knocked it into 1b. If you look at the primordium here in magenta, and the integrin beta 1b, the endogenous locus labeled, you see that the primordium, actually all the cells are making integrin beta 1b, okay? And you also see that the muscle are making it here. So this is a muscle cell. And also on top of it here, you see that the skin cells are making it, okay? So you have integrin beta 1b in a lot of the tissues. Okay, so that would argue you could use, or this structure could use integrins to couple force to the outside. To test that what we did is we just used an integrin beta 1b mutant. So we removed it from all of the entire embryo and asked what happens to the migration of the tissues. So this is a 2-day-old embryo, and you see in wild type the entire structure is getting to the tip of the tail, is depositing the sensory organs. In a integrin beta 1b mutant, you actually have slowed migration and the primordium does not reach the tip of the tail. But, and that's important, and we will see probably why later, it still can migrate, right? It's not completely blocked. Okay, so this is a live movie of this. So you see a wild type or heterozygous embryo where the primordial migration is fine. If you mutate integrin beta 1b in the entire embryo, the primordium is not migrating at the same speed. So it's less efficient in its migration. Okay. In this experiment, what we do is we take integrin beta 1b away in all the cells of the embryo, right? Whatever cell makes it can't make it because we deleted the gene. Okay, so we wanted to know whether deleting integrin beta 1b only in the migrating tissue would also cause a migration phenotype. So is it required in the migrating tissue only, or is it a function of having integrin beta 1b in the skin and the muscle and other tissues, right? So the way to do this, what we did is we used a diagram approach. So this diagram we developed and we called it ZGRAD. We adapted it from flies. What it does is you express a nanobody against GFP coupled to an F box that will recruit the nanobody to a E3 ubiquitin ligase. The nanobody will bind to GFP, and whatever you fuse to GFP, your protein of interest, will then also be degraded. Okay, so that's the diagram approach. So we took this and wanted to place this diagram into the migrating structure only. So we used a promoter, CXCR4, the chemokine receptor, to express it in the primordium in a fish that had integrin beta 1b tagged with GFP. Okay, so integrin beta 1b is in most of the fish. If you now put in ZGRAD only in the migrating structure, what you will do is you will deplete GFP, integrin beta 1b GFP, only in the migrating tissue. Okay, so you have a tissue-specific depletion of your protein. So when you do this, this actually works. So this is the migrating tissue, integrin beta 1b and B in green. And here we are expressing the diagram system in the primordium. And if you then take away the primordial marker, you see that you deplete integrin beta 1b. So now you have a tissue-specific loss of function or reduction in function. And when you do this, as the same as when you deplete integrin beta 1b in the entire fish, again, you are slowing down the migration. Okay. And actually, in this fish, what we also did is we removed the other integrin beta 1a. So that's basically telling us that you need integrin beta 1 to efficiently migrate. Okay. So not going to talk much about the TALEN experiments, but there's a nice way of actually disrupting the TALEN binding and another factor, kindlin, to integrins. And what you can do is you can delete the binding sites for these proteins in the tail of integrin beta 1b. And we did this. We went in and deleted that in the gene and still put a GFP on at the endogenous locus. And the first thing you see is here, this is the primordium cross-section. This is the front. Okay, my mouse stopped working. Okay, this is the back. In green, you have integrin beta 1b. And in the wild type version, the one that can bind to TALEN and kindlin, you see nice enrichment of integrin beta 1b. If you delete the MPXY motif, the binding domains, you lose that. So integrin beta 1b's are not localized properly anymore. And again, if you look at migration, you get the same thing. The primordium is migrating slower and does not get to its destination. Okay, so that would argue you probably also need the interaction to TALEN and kindlin for efficient migration. Okay, so one idea in cell culture and also in animal studies is that integrins couple actin flow to the outside, right? So we were wondering whether this happens in our cells too. I showed you initially that row A is required to have a lot of actin flow, actin dynamics in the cells in the primordium. So what we did is we looked at a single cell. So we labeled a single cell in the migrating tissue in wild type and a single cell in the migrating tissue in the primordium in an integrin beta 1b mutant. and then we looked at how actin is behaving in an individual cell. So when you do this, what you find is that in wild type, you have a lot less actin flow than in an integrin beta 1b mutant. And you can quantify this by using a chymograph analysis. So you see basically here the cell is moving forward and actin is pretty much constant at the position of the cell. In the beta 1b, you have a lot more slippage and actually the flow rate goes up. Okay, so that would argue that integrin beta 1b in the primordium is required to couple the actin flow in the cells to the outside, to the substrate. Okay, so that would probably be the force coupling, like your, the sole of your feet when you move, when you walk on the on the beach. So that would be coupling. And the last part, what I want to talk about is how the force that you're generating in the cells, that is partly coupled through integrins to the outside, how that is pushing and pulling on the substrate, on the basement membrane. So in cell culture, the way people do this is they put a cell on a polyacrylamide gel with known physical properties. So the stiffness and something called the Poisson ratio you need to know. And then what you do is you basically measure the displacement of these beads, and by knowing the displacement and the physical properties of the polyacrylamide gel, you can infer how much pulling and pushing the cell is doing on the substrate. So we wanted to do that, not in cell culture, but in the embryo. So it's much harder for us to get these beads in there. So the way we thought about doing this is somebody is drawing blue lines on the screen here. So the way we did this is we had the basement membrane labeled, right, already with laminin. So we decided to use that, and instead of putting beads into the embryo, what we're doing is we're putting bleached dots in front of the primordium, right, in a certain pattern. So we bleach a dot, and these dots are then unique, and we can see how they're being pushed and how they're being displaced as cells are crawling across them. So that was our beads. The physical properties of the substrate of the basement membrane we needed to measure, and I'm going to show you that we used atomic force microscopy to do that. And if you combine these two, then you have the same thing as in cell culture. You can actually back calculate the stresses that give rise to the deformation pattern of your bleached dots on the basement membrane. Okay, so that is the idea. So here is a movie. You have the muscle segments here in dark green. In light red, you have the primordium. So it's going to migrate from here all the way to here. And in front of this, of the primordium, of this tissue, we placed a pattern of bleached dots. So the green is a component of the basement membrane, the laminin C1. And I'm not going to show you that, but this is actually fairly stably incorporated and does not diffuse much over a couple of hours. Okay, so if you play this movie, what you're going to see is the tissue moving through this pattern of bleached dots. Okay, so what you now need to do is you need to now trace the displacement of all these dots. To do that, we teamed up with the computational science department here at NYU. And what they did is they developed a software that is tracking all these dots over time. And that will then allow us to basically have for each dot its deformation in space and time. With that, you basically have the displacement. And in principle, if you know the physical properties, you can back calculate to how much force you need to generate this displacement. So this comes back to something we've learned in high school, and that's the Hooke's law, that force is equal to some constant times the displacement, right? So this is basically, not sure whether you can see, that's the rubber band here. Two dots are on there. So those two dots are basically the dots on the basement membrane. And if I pull on the rubber band, right, and if I know the stiffness or the spring constant of the rubber band, and I measure the displacement of these two dots over time, I can infer how much pulling I do on this rubber band, okay? So this is one of the experiments I'm doing in this talk. This is basically what we're doing. This is for one dimension, right? So in 3-dimension, Hooke's law gets a little bit more complicated. You need to know the stiffness, which is called also Young's modulus, but you also need to know something called the Poisson ratio. And the Poisson ratio basically describes if you have like a piece of Play-Doh or any material, and you pull or push on it. If I push on it, it expands sideways, right? So the Poisson ratio tells you how much when I push, how much that material is going to go expand in X, Y, right? In sideways. So we can't measure that. So we just assumed that the basement membrane is roughly isotropic and behaves like a rubber band, which gives you a Poisson ratio, ideally of 0.5. We used 0.45. But the stiffness we can measure. So we needed to get to the basement membrane, and we're going to use atomic force microscopy. Just a brief intro how that works. You have a cantilever that that has a little reflective surface here. You shine a laser on it, and the laser is being reflected, and you are recording the reflection of the laser. And as you lower the cantilever, at some point you're touching your material. The cantilever is bending. The bending, you know the spring constant for this bending of the cantilever, and you're monitoring at the same time how much bending there is using the laser. And as you lower it, as you go onto the material, the stiffer it is, the more bending you get. So you have basically a readout of the stiffness of your material. Okay. So basically, this is the approach curve here. You go in, hit the material, and then you're going to get the resistance. And depending on how much resistance you get, that using a certain model, you can infer the stiffness of the material that you're touching. Okay, so that's what we did as we went in with a laser, with a cantilever here, and peeled off the skin to get to the basement membrane and did these measurements. So this is what it looks like in real life. This is a control embryo, the skin labeled with equaterin in magenta, the basement membrane and with LAMC1 again. And then we peeled off the skin and used an atomic force microscope to collect these approach curves. So we're going in here with the cantilever, hitting the basement membrane, getting resistance, and we're modeling this piece of the approach curve to basically get the stiffness, the spring constant for the basement membrane. Did this a lot, so you get a lot of measurements, and we get something about 600 Pascal as Young's modulus. Okay, and with that and the deformation, when we put that into the software, we can calculate the stresses. So what I'm going to first show you is the displacement of the basement membrane as the tissue is crawling. So this is basically just looking at how much a dot is moving during the movie. So what you're going to see is the primordium is migrating in from here. We are actually positioned on the muscle side. So we're looking at the basement membrane from underneath. The primordium is on top, and the skin would be even outside. Okay, so what is going to happen is this is playing. Let me see.
No.
Okay, no.
Oh, yeah, now it's playing.
The tissue is moving. The very front here is displacing the basement membrane very little. So you see these vectors that are indicating the displacement. So the magnitude is color-coded, and the direction you can see just by where it points to, right? So you have very little displacement in the front and a lot more displacement on the sides of the structure and actually in the back of the structure, okay? So this is the displacement. Then with that data, you can extract all kinds of stresses. One thing you can extract is the traction stresses. You can imagine like you walking across a rubber sheet like a trampoline, and as you walk, right, you're going to indent the rubber sheet. And if you look at the basement membrane as a rubber sheet, and we ask how much are the cells that are crawling across at this tissue, how much is it pushing down in terms of pressure, force per area, you're going to see this here in this movie. So this is basically one way to look at it. And you see that there's very little pushing stresses here in the front, and there's a lot more on the side and more in the back. Okay, so that's one way of looking at it. Another way of looking at it is asking how much are these cells pushing and pulling on the basement membrane in the direction of migration, right? So how much are they pulling and pushing along the way they walk? And this is depicted here. So you're going to have, again, the tissue, and you're going to have these arrows indicating now the stresses in the direction of migration. Okay, so if you look at this, you're going to see, again, very little and not very directional arrows here in the front of the tissue, and you're going to see a lot more pushing and pulling on the sides and in the back. So what this argues is that these cells are actually doing very little pushing and pulling and very unidirectional, not directional in the front, but much more on the sides and in the back. So there's more force generated in the back to do this, right? So one thing that people have done a long time ago to look at these kind of traction or stresses that cells are generating on a substrate is called wrinkle microscopy. So what you have is, you think of this as a basement membrane here, and if you crawl on it and you pull on it, right, you create wrinkles, and these wrinkles will increase the intensity of your signal of the basement membrane. So we were asking whether the primordium, as it moves across the basement membrane and pushes on it, whether it actually creates wrinkles. And we would see those wrinkles by the basement membrane being concentrated at these areas, right? So that's what we did. This is a live primordium, just marked. And here you have pseudo-colored the intensities of the basement membrane. And you can see that you have a lot more basement membrane concentrations on the edges of the structure and in the back, arguing that you create wrinkles there. And that is corresponding to where we see most of the myosin activity on the edges and also more in the back. So that would argue, consistent with the stresses, that you push and pull more on the sides and the back than in the front. Okay, and to summarize, the stresses that you see, you have kind of like a press stroke like movement. You have very little pushing, pulling in the front, but a lot more on the sides and in the back. So, and this brings me to the model of how we think these cells are generating the motility. You have, within the cell, you have actin flow that you partly couple through integrins to the basement membrane. So if you don't have that, you slow down in migration. In response to that, you upregulate your actin flow, actually. And you have this in all cells, front and back. But the back cells are pushing and pulling harder, especially the ones on the side, than the front cells, and with more directionality. And on a tissue level, that gives you more deformations, pushing, pulling in the back than the front. So this actually probably helps the front push forward and wedge between the skin and the muscle. And with that, I would like to thank the two people who did the work. So Naoya did most of the biology, and Z did most of the computational stuff. And this was not in empty space. This was done at NYU in my lab, and we are basically sharing the lab with another fish person, Jesus Torres Vasquez, a colleague of mine. So that's the biology side, and then Z was guided by a collaborator, Daniela Panozzo, at the computational science unit, and helped by a postdoc, Teseo Schneider. Okay, and with that, I'm happy to take any questions.
Awesome. Thank you so much. We'll go to the questions in a second. I don't know if you want to keep your screen up or if you want to stop the share. But I just want to say, first of all, I teach a cell migration class in the spring, and I'm so excited to use your props that you showed because they're really, really good. So thank you for doing those. And I'm really excited to read this paper in my class in the spring because it's so cool. And the technique of making dots to be able to do in vivo traction force microscopy is just, it's so clever. I'm dying over here. So I just wanted to compliment you guys on that because it's really cool. So I have a question, but let's let Anna go first. Go ahead, please.
So, Holger, I just want to say this is just elegant, beautiful, awesome science. So thank you.
Well, thank you. I mean, and thank Naoya. I mean, he did it, right? Not me.
Yeah. So what happens with the beta-1 integrin depletion with those traction forces? Have you guys done that? Because it's striking that with the beta-1 integrin depletion, those cells are migrating really very well. And so
we haven't done it with the depletion. We have done it only in the integrin beta-1b mutant global mutant, because the depletion, there's a lot of things genetically we need to bring together. But if you actually look at the beta integrin, so if you take beta-1b away and you look at traction, you lower the traction. And pretty much the same in the front and the back. So it goes down everywhere.
Yeah, it's interesting.
So you couple less, but you still couple, right? And you still move.
Yeah, I mean, so it's interesting. So you have laminin on one side. What's on the other side? Is there
the skin?
So it is. So the skin is probably, so it's some compression, do you think? Compression
definitely happens. What we did is, so one idea was like the skin is also doing something, right? So, and you have E-cadherin expressed in the primordium and N-cadherin, and the skin is making E-cadherin. So we were asking whether if we impair cell-cell adhesion by depleting alpha-catenin in the primordium only, using again the stagron system, whether we would also slow it down, right? Because maybe there's some coupling to the top too, to the skin. And we didn't see a slowing down when we removed alpha-catenin in the primordium. What does happen is they get less directional, the cells, and the deposition of the sensory organs is impaired, because you need a lot of adhesion to cluster those. Yeah.
Well,
but remember, but in all of that, we are depleting. We're not killing everything, right? So this is always a caveat.
Yes, right. Yes. Well, thank you so much.
Thank you, Holger. There was initially a question from Bob asking, is there some sort of protease activity required to separate the basement membrane from the skin layer ahead of the migrating front?
Mm-hmm. Yeah, we don't know. It's a good one, but but we don't know.
Okay, perfect.
We suspect for different reasons that there is some kind of activity like that, but that would,
separate it.
Yeah, and also, the chemokine probably needs some separation too. Yeah.
Cool. We can take the next question from Mike, if you want to go ahead.
That was really amazing to do all these traction experiments in vivo. Actually, my first question you already answered. I wanted to know if there's like E-cadherin between the skin and the migrating cells, because it reminded me a bit of border cell migration, which basically migrates only with E-cadherin. And not on ECM. My other question was, do you believe that actin polymerization plays no role in the retrograde flow? Do you think it's solely the row-rock myosin axis, or is there also kind of lamellopodia in the front where we have actin polymerization? Because you said, CDC42 and REC don't seem to be that important.
Okay, so I think actually it's a combination of the two, but when we block Rho, there's still some dynamics of actin going on, but very little, right? So, and we do not see much protrusive activity when we do this. It's kind of like you're freezing them. If you have a protrusion, that protrusion is still there, but it doesn't retract or extend anymore. So that's as much as I can say to that. I would be very surprised if you completely remove RAC or CDC42 that you do not get some kind of phenotype, but we haven't done that. We were going off by what is the polarity, right? I mean, would we see a difference in RAC polarization or CDC42 polarization? And we didn't see that, and that's why we focus on Rho. It's because Rho we do see asymmetries within this.
And then a quick question, just a technical thing about the sensor you used for Rho. Was that like an RBD-based sensor, or?
Yeah, so this is that's a good one. So for Rho A, we're using HPH, so in a Nillen domain, and we've made also the RGBD sensor, and they're partly overlapping, partly the HPH is more concentrated than a GBD-based sensor.
Okay, thank you very much.
Awesome. There was another question from Bob, which I missed. It was later in the chat, asking, are there any traction force applied in the skin layer? I also had a similar question because looking at the skin and the muscle, are the traction forces sort of different on the top versus bottom?
We can only look at the traction forces on the basal membrane. So we are lucky in that sense. I mean, there are many things that made this possible. One of the things, laminin doesn't diffuse, right? If it was diffusive, we couldn't have done this. There is no equivalent of something like the basal membrane on the skin, right? It would need to be something that is not moving, right, on its own. So we don't have that. So we cannot, the only thing you could do is model. That's, but we can't, I can't ask, all we see is the basal membrane.
Awesome, thank you. Ajay, if you want to go ahead.
Great
Hi.
talk, Holger. I had a question about how to interpret the fact that you see more traction at the back. And one interpretation of that is that you have something that's moving and it's just literally rear wheel drive. But another way of thinking about it is that you have a column of cells that is moving, and that to keep that column of cells moving coherently, the forces that need to be applied from the front to the back are somewhat different. And that if you don't apply more force from the back, it'll start stretching out. And to keep it all going together, you actually have this more force from the back. So it's not literally rear wheel drive as if the front is just a plow that is pushing through with the back driving this thing. But it's an emergent property of the differential requirements along the column of the system. And that allows the whole thing to move properly. Do you think that's a reasonable alternate or explanation? Because I felt the paper was sort of emphasizing it's literally rear wheel drive, which...
I hope we didn't quite state it like that in the paper, because I don't think that's true. And the best experiment to do this is if you make a cell in the front or a couple of cells in the front non-directional, right? So you take CXCR4 A and B away in them, the clone, it basically jams, right? So what you have is back pushing against it. So it's not, back is not enough to plow through the skin and muscle. So you cannot do this. So you need the front to move too.
Right.
So this is why a rear engine is not, you need a combination of the two.
Correct.
And it could be what you're saying that, so what you're referring to is basically you have mesenchymal cells in the front and they get more epithelial in the back, right? So they're more columnar, right? That's what you're referring to. And that difference might actually require different forces. And one force is that you need to do is you need to actually push the muscle and the skin further apart in the back than in the front. But definitely, I mean, let's be clear. I mean, I don't mean to say that the rear is like, it's not like a VW van where you have a rear engine and the front doesn't do anything. You need the front to collaborate here.
No, beautiful work. And I also especially like your strategy for doing the traction. That was just like pure brilliance. Wonderful. Thanks a lot.
And I want to put in a quick plug for everyone here. Ajay will be presenting in about a month. So if you're interested in the lateral line and in zebrafish, please come back for that talk. Next up, Megan, please go ahead.
Hi. Oh, wait, can you hear me? Oh, hi. I'm Megan. I'm one of Ajay's postbacks. So I actually have done some modeling with the primordium. And I just wanted, so one of the papers that we looked at specifically was by Damien, and it says how the superficial cells are necessary for proper migration within the primordium itself. So I wanted to see if you've done any work looking at the traction of those superficial cells and how they relate to the overall, how they relate to tractional forces within the migrating primordium.
Yeah, so this basically gets back to the alpha-catenin experiment where we're trying to disrupt like catenin-mediated adhesion between the primordium and the skin, where we did not see a contribution to the speed of migration. The other evidence we have is if we look at F-actin dynamics in the superficial cells versus the basal side of the primordium, we on the top, we do not see a difference when we fiddle with integrin beta 1b. So that's as much as can speak to that. But I mean, this is, I mean, from your lab, right? I mean, in Chitness's lab, right?
Yeah.
So you have this Eli paper where you basically peeled away the skin, right, and that blocked migration. So the skin definitely does something. Maybe it's compression, maybe it's also part of the substrate. I'm not trying to say it's not, right, but we have no evidence for it.
Of course, thank you so much.
Next question, Harlan, you can please go ahead.
Hello, Holger.
Hi.
When you look at the pulling and pushing forces, obviously you do not have cellular resolution there, because what you see is these changes and the cells that are overlying those areas. Nevertheless, did you look, and if so, did you find any correlation of the shape of the cells and the pulling and pushing? And if so, can you now use those shapes as a proxy for what the cells are doing?
We haven't done that. And one thing we tried, so the resolution is determined, the resolution we get is determined by how closely the marks are, right? So so you make a pleach start, and you can basically only, I mean, the modeling then actually infers like things in between. You can go, it's gonna be continuous, but you have discrete measurements basically at each pleach dot. So if you make them closer, if you decrease the spacing, you get better resolution. So we've been trying to do this using 2 microns, which gets you something closer to like a subcellular resolution, but then the tracking becomes a lot more difficult. The software, I mean, this is a lot of tracking, right? If the software doesn't do that for you, you spend months doing this. So yeah, but basically the short answer is I cannot give you a correlation between shape and forces or stresses.
Thank you.
Okay, fantastic. If more people want to drop any questions in the chat or raise their hand, please go ahead. I have a question, and this might be a little bit too basic, but when you showed the mRNA expression of the chemokine, the SDF1, it looked like it was, and again, you know, maybe this isn't inferring too much, but it looked like it was pretty consistently expressed all the way along the path. So do you think that there's any like self-generated gradient as the cells are moving along and consuming it, or how is the gradient really formed to get them to be unidirectional in migration? And again, that may just be ignorance on my part on how the system works.
So you're right. So the back tissue makes a clearance receptor for the chemokine, and that generates a gradient across the tissue that it moves with. So you're completely right. I mean, we haven't done it thoroughly, but we quantified the chemokine fused to GFP at different positions, and we don't see a difference, right? So it's pretty uniform, and the only thing that makes a gradient is then having a clearance receptor only in the back of the migrating tissue that makes you a self-generated gradient and propels you forward.
Okay, thanks for explaining that. Yeah, that's really interesting, right? Because a lot of us think about chemo attraction as happening a source and a sink, but the cells are in some sense making their own gradient just by clearing it differentially across the tissue spatially. Is that right?
Mm-hmm.
That's so
what
a really cool system. My goodness. It's really neat. Sorry, go ahead.
I think Mike has another question. Yeah.
Mike, go ahead.
Yeah, it's also a bit of a basic question because I'm in terms of PLA person as a Drosophilus. Is there any motion among the cells, or do cells which are mesenchymal in the front always stay mesenchymal cells? Because there was this talk about rear wheel drive, and of course I thought about this by your paper where you have like these swirls going on.
So you're asking whether the cells in the front, like during the migration,
yeah,
there's very little mixing of the cells. Is that what you're asking? So
yeah,
a front cell hardly ever, I mean, they change positions a little. So it's not like the leader cell is always in the front. They can change a little, but most of the cells, if you track them, they're amazingly persistent in their migration.
Okay.
Unless you fiddle with, I mean, you know, you can make them do things, but in wild type they're not. The front cells at the very end, right, when they get to the tip of the tail, they will differentiate and they will become epithelial, right? So then it's different. Like when they stop migrating, they make their last sensory organs, then yeah, that will change.
Okay, thank you.
Okay, great. Thanks so much. That was perfect timing. So thank you for leaving so much time for questions, Holger. That was really fantastic. I'm going to go ahead and sign off from our recording. And if anyone has any last minute questions, y'all can bring those in now. But for those of you watching on YouTube, thanks so much.